The polymerase chain reaction is a biochemical technology in molecular biology used to amplify a single or a few copies of a piece of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. Developed in 1983 by Kary Mullis, PCR is now a common and often indispensable technique used in medical and biological research labs for a variety of applications. These include DNA cloning for sequencing, DNA-based phylogeny, or functional analysis of genes. The diagnosis of hereditary diseases. The identification of genetic fingerprints. And the detection and diagnosis of infectious diseases. In 1993, Mullis was awarded the Nobel Prize in Chemistry along with Michael Smith for his work on PCR. The method relies on thermal cycling, consisting of cycles of repeated heating and cooling of the reaction for DNA melting and enzymatic replication of the DNA. Primers containing sequences complementary to the target region along with a DNA polymerase are key components to enable selective and repeated amplification. As PCR progresses, the DNA generated is itself used as a template for replication, setting in motion a chain reaction in which the DNA template is exponentially amplified. PCR can be extensively modified to perform a wide array of genetic manipulations. Almost all PCR applications employ a heat-stable DNA polymerase, such as Tack Polymerase. This DNA polymerase enzymatically assembles a new DNA strand from DNA building blocks, the nucleotides, by using single-stranded DNA as a template and DNA oligonucleotides, which are required for initiation of DNA synthesis. The vast majority of PCR methods use thermal cycling, that is, alternately heating and cooling the PCR sample through a defined series of temperature steps. In the first step, the two strands of the DNA double helix are physically separated at a high temperature in a process called DNA melting. In the second step, the temperature is lowered and the two DNA strands become templates for DNA polymerase to selectively amplify the target DNA. The selectivity of Pcr results from the use of primers that are complementary to the DNA region targeted for amplification under specific thermal cycling conditions. Pcr principles and procedure. Pcr is used to amplify a specific region of a DNA strand. Most Pcr methods typically amplify DNA fragments of between 0.1 and 10 kilobase pairs, although some techniques allow for amplification of fragments up to 40 kilobytes in size. The amount of amplified product is determined by the available substrates in the reaction, which become limiting as the reaction progresses. A basic Pcr setup requires several components and reagents. These components include DNA template that contains the DNA region to be amplified. Two primers that are complementary to the 3' ends of each of the sense and antisense strand of the DNA target. Tack Polymerase or another DNA polymerase with a temperature optimum at around 70 °C. Deoxynucleoside trophosphates, the building blocks from which the DNA polymerase synthesizes a new DNA strand. Buffer solution, providing a suitable chemical environment for optimum activity and stability of the DNA polymerase. Bivalent cations, magnesium or manganese ions. Generally Mg2+ is used, but Mn2+ can be utilized for PCR-mediated DNA mutagenesis, as higher Mn2+ concentration increases the error rate during DNA synthesis. Monovalent cation potassium ions. The Pcr is commonly carried out in a reaction volume of 10 200 Ai1/4 L in small reaction tubes in a thermal cycler. The thermal cycler heats and cools the reaction tubes to achieve the temperatures required at each step of the reaction. Many modern thermal cyclers make use of the Peltier effect, which permits both heating and cooling of the block holding the Pcr tubes simply by reversing the electric current. Thin-walled reaction tubes permit favorable thermal conductivity to allow for rapid thermal equilibration. Most thermal cyclers have heated lids to prevent condensation at the top of the reaction tube. Older thermocyclers lacking a heated lid require a layer of oil on top of the reaction mixture or a ball of wax inside the tube. Procedure: typically, Pcr consists of a series of 20 to 40 repeated temperature changes, called cycles, with each cycle commonly consisting of 2 to 3 discrete temperature steps, usually 3. The cycling is often preceded by a single temperature step at a high temperature, and followed by one hold at the end for final product extension or brief storage. The temperatures used and the length of time they are applied in each cycle depend on a variety of parameters. These include the enzyme used for DNA synthesis, the concentration of divalent ions and DNTPS in the reaction, and the melting temperature of the primers. Initialization step. This step consists of heating the reaction to a temperature of 94°-96 °C, which is held for 1°-9 minutes. It is only required for DNA polymerases that require heat activation by hot start PCR. Denaturation step. This step is the first regular cycling event and consists of heating the reaction to 94°-98 °C for 20°-30 seconds. It causes DNA melting of the DNA template by disrupting the hydrogen bonds between complementary bases, yielding single-stranded DNA molecules. Annealing step. The reaction temperature is lowered to 50°-65 °C for 20°-40 seconds, allowing annealing of the primers to the single-stranded DNA template. Typically the annealing temperature is about 3°-5 °C below the Tm of the primers used. Stapled DNA hydrogen bonds are only formed when the primer sequence very closely matches the template sequence. The polymerase binds to the primer template hybrid and begins DNA formation. Extension elongation step. The temperature at this step depends on the DNA polymerase used. Tack Polymerase has its optimum activity temperature at 75°-80 °C, and commonly a temperature of 72 °C is used with this enzyme. At this step the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding DNTPS that are complementary to the template in 5' to 3' direction, condensing the 5-phosphate group of the DNTPS with a 3-hydroxyl group at the end of the nascent DNA strand. The extension time depends both on the DNA polymerase used and on the length of the DNA fragment to be amplified. As a rule of thumb, at its optimum temperature, the DNA polymerase will polymerize 1,000 bases per minute. Under optimum conditions, that is, if there are no limitations due to limiting substrates or reagents, at each extension step, the amount of DNA target is doubled, leading to exponential amplification of the specific DNA fragment. Final elongation. This single step is occasionally performed at a temperature of 70°-74 °C for 5°-15 minutes after the last Pcr cycle to ensure that any remaining single-stranded DNA is fully extended. Final hold. This step at 4°-15 °C for an indefinite time may be employed for short-term storage of the reaction. To check whether the Pcr generated the anticipated DNA fragment, Agarose Gel Electrophoresis is employed for size separation of the Pcr products. The size, S, of Pcr products is determined by comparison with a DNA ladder, which contains DNA fragments of known size, run on the gel alongside the Pcr products. Pcr stages. The Pcr process can be divided into 3 stages. Exponential amplification. At every cycle, the amount of product is doubled. The reaction is very sensitive, only minute quantities of DNA need to be present. Leveling off stage. The reaction slows as the DNA polymerase loses activity and as consumption of reagents such as DNTPS and primers causes them to become limiting. Plateau. No more product accumulates due to exhaustion of reagents and enzyme. Pcr optimization. In practice, Pcr can fail for various reasons, in part due to its sensitivity to contamination causing amplification of spurious DNA products. Because of this, a number of techniques and procedures have been developed for optimizing Pcr conditions. Contamination with extraneous DNA is addressed with lab protocols and procedures that separate pre-Pcr mixtures from potential DNA contaminants. This usually involves spatial separation of Pcr setup areas from areas for analysis or purification of Pcr products, use of disposable plasticware, and thoroughly cleaning the work surface between reaction setups. Primer design techniques are important in improving PCR product yield and in avoiding the formation of spurious products, and the usage of alternate buffer components or polymerase enzymes can help with amplification of long or otherwise problematic regions of DNA. Addition of reagents, such as formamide, in buffer systems may increase the specificity and yield of PCR. Computer simulations of theoretical PCR results may be performed to assist in primer design. Application of Pcr. Selective DNA isolation. Pcr allows isolation of DNA fragments from genomic DNA by selective amplification of a specific region of DNA. This use of Pcr augments many methods, such as generating hybridization probes for southern or northern hybridization and DNA cloning, which require larger amounts of DNA, representing a specific DNA region. Pcr supplies these techniques with high amounts of pure DNA, enabling analysis of DNA samples even from very small amounts of starting material. Other applications of Pcr include DNA sequencing to determine unknown Pcr amplified sequences in which one of the amplification primers may be used in Sanger sequencing, isolation of a DNA sequence to expedite recombinant DNA technologies involving the insertion of a DNA sequence into a plasmid or the genetic material of another organism. Bacterial colonies can be rapidly screened by Pcr for correct DNA vector constructs. Pcr may also be used for genetic fingerprinting, a forensic technique used to identify a person or organism by comparing experimental DNAs through different Pcr-based methods. Some Pcr fingerprints methods have high discriminative power and can be used to identify genetic relationships between individuals, such as parent-child or between siblings, and are used in paternity testing. This technique may also be used to determine evolutionary relationships among organisms. Amplification and quantification of DNA. Because Pcr amplifies the regions of DNA that it targets, Pcr can be used to analyze extremely small amounts of sample. This is often critical for forensic analysis, when only a trace amount of DNA is available as evidence. Pcr may also be used in the analysis of DNA that is tens of thousands of years old. These Pcr-based techniques have been successfully used on animals, such as a 40,000-year-old mammoth, and also on human DNA, in applications ranging from the analysis of Egyptian mummies to the identification of a Russian Tsar and the body of English King Richard III. Quantitative Pcr methods allow the estimation of the amount of a given sequence present in a sample. A technique often applied to quantitatively determine levels of gene expression. Quantitative Pcr is an established tool for DNA quantification that measures the accumulation of DNA product after each round of Pcr amplification. Pcr in diagnosis of diseases. Pcr permits early diagnosis of malignant diseases such as leukemia and lymphomas, which is currently the highest developed in cancer research and is already being used routinely. Pcr assays can be performed directly on genomic DNA samples to detect translocation-specific malignant cells at a sensitivity that is at least 10,000-fold higher than that of other methods. Pcr allows for rapid and highly specific diagnosis of infectious diseases, including those caused by bacteria or viruses. Pcr also permits identification of non-cultivatable or slow-growing microorganisms such as mycobacteria, anaerobic bacteria, or viruses from tissue culture assays and animal models. The basis for Pcr diagnostic applications in microbiology is the detection of infectious agents and the discrimination of non-pathogenic from pathogenic strains by virtue of specific genes. Viral DNA can likewise be detected by Pcr. The primers used need to be specific to the targeted sequences in the DNA of a virus, and the Pcr can be used for diagnostic analyses or DNA sequencing of the viral genome. The high sensitivity of Pcr permits virus detection soon after infection and even before the onset of disease. Such early detection may give physicians a significant lead time in treatment. The amount of virus in a patient can also be quantified by Pcr-based DNA quantitation techniques. Variations on the basic Pcr technique. Allele-specific Pcr, a diagnostic or cloning technique based on single nucleotide variations. It requires prior knowledge of a DNA sequence, including differences between alleles, and uses primers whose 3' ends encompass the SNV. Pcr amplification under stringent conditions is much less efficient in the presence of a mismatch between template and primer, so successful amplification with an SNP-specific primer signals presence of the specific SNP in a sequence. See SNP genotyping for more information. Assembly Pcr polymerase cycling assembly. Artificial synthesis of long DNA sequences by performing Pcr on a pool of long oligonucleotides with short overlapping segments. The oligonucleotides alternate between sense and antisense directions, and the overlapping segments determine the order of the Pcr fragments, thereby selectively producing the final long DNA product. Asymmetric Pcr preferentially amplifies one DNA strand in a double-stranded DNA template. It is used in sequencing and hybridization probing where amplification of only one of the two complementary strands is required. Pcr is carried out as usual, but with a great excess of the primer for the strand targeted for amplification. Because of the slow amplification later in the reaction after the limiting primer has been used up, extra cycles of Pcr are required. A recent modification on this process, known as linear after the exponential Pcr, uses a limiting primer with a higher melting temperature than the excess primer to maintain reaction efficiency as the limiting primer concentration decreases mid-reaction. Dial Out Pcr, a highly parallel method for retrieving accurate DNA molecules for gene synthesis. A complex library of DNA molecules is modified with unique flanking tags before massively parallel sequencing. Tag-directed primers then enable the retrieval of the molecules with desired sequences by Pcr. Digital Pcr, used to measure the quantity of a target DNA sequence in a DNA sample. The DNA sample is highly diluted so that after running many Pcrs in parallel, some of them will not receive a single molecule of the target DNA. The target DNA concentration is calculated using the proportion of negative outcomes. Hence the name digital Pcr. Helicase-dependent amplification, similar to traditional Pcr, but uses a constant temperature rather than cycling through denaturation and annealing extension cycles. DNA helicase, an enzyme that unwinds DNA, is used in place of thermal denaturation. Hot start Pcr, a technique that reduces non-specific amplification during the initial setup stages of the Pcr. It may be performed manually by heating the reaction components to the denaturation temperature before adding the polymerase. Specialized enzyme systems have been developed that inhibit the polymerase's activity at ambient temperature either by the binding of an antibody or by the presence of covalently bound inhibitors that dissociate only after a high-temperature activation step. Hot start cold finish Pcr is achieved with new hybrid polymerases that are inactive at ambient temperature and are instantly activated at elongation temperature. In silico Pcr refers to computational tools used to calculate theoretical polymerase chain reaction results using a given set of primers to amplify DNA sequences from a sequenced genome or transcriptome. In silico Pcr was proposed as an educational tool for molecular biology. Intersequence-specific Pcr, a Pcr method for DNA fingerprinting that amplifies regions between simple sequence repeats to produce a unique fingerprint of amplified fragment lengths. Inverse Pcr, is commonly used to identify the flanking sequences around genomic inserts. It involves a series of DNA digestions and self-ligation, resulting in known sequences at either end of the unknown sequence. Ligation-mediated Pcr, uses small DNA linkers ligated to the DNA of interest and multiple primers annealing to the DNA linkers. It has been used for DNA sequencing, genome walking, and DNA footprinting. Methylation Specific Pcr, developed by Stephen Balin and Jim Herman at the Johns Hopkins School of Medicine, and is used to detect methylation of CpG islands in genomic DNA. DNA is first treated with sodium bisulfate, which converts unmethylated cytosine bases to uracil, which is recognized by Pcr primers as thymine. two PCRs are then carried out on the modified DNA, using primer sets identical except at any CpG islands within the primer sequences. At these points, one primer set recognizes DNA with cytosines to amplify methylated DNA, and one set recognizes DNA with uracil or thymine to amplify unmethylated DNA. MSP using qPCR can also be performed to obtain quantitative rather than qualitative information about methylation. Miniprimer PCR uses a thermostable polymerase that can extend from short primers as short as 9 or 10 nucleotides. This method permits PCR targeting to smaller primer binding regions, and is used to amplify conserved DNA sequences, such as the 16S rRNA gene. Multiplex ligation-dependent probe amplification permits multiple targets to be amplified with only a single primer pair, thus avoiding the resolution limitations of multiplex PCR. Multiplex PCR consists of multiple primer sets within a single PCR mixture to produce amplicons of varying sizes that are specific to different DNA sequences. By targeting multiple genes at once, additional information may be gained from a single test run that otherwise would require several times the reagents and more time to perform. Annealing temperatures for each of the primer sets must be optimized to work correctly within a single reaction, and amplicon sizes. That is, their base pair length should be different enough to form distinct bands when visualized by gel electrophoresis. Nanoparticle Assisted Pcr. In recent years, it has been reported that some nanoparticles can enhance the efficiency of PCR, and some even perform better than the original PCR enhancers. It was also found that quantum dots can improve PCR specificity and efficiency. Single-walled carbon nanotubes and multi-walled carbon nanotubes are efficient in enhancing the amplification of long PCR. Carbon nanopowder was reported to be able to improve the efficiency of repeated PCR and long PCR. ZNO, TO2, and AgNPs were also found to increase PCR yield. Importantly, already known data has indicated that non-metallic NPs retained acceptable amplification fidelity. Given that many NPs are capable of enhancing PCR efficiency, it is clear that there is likely to be great potential for nanope technology improvements and product development. Nested PCR increases the specificity of DNA amplification by reducing background due to non-specific amplification of DNA. Two sets of primers are used in two successive PCRs. In the first reaction, one pair of primers is used to generate DNA products, which besides the intended target, may still consist of non-specifically amplified DNA fragments. The product, S, are then used in a second PCR with a set of primers whose binding sites are completely or partially different from and located 3 feet of each of the primers used in the first reaction. Nested PCR is often more successful in specifically amplifying long DNA fragments than conventional PCR, but it requires more detailed knowledge of the target sequences. Overlap extension Pcr or splicing by overlap extension, a genetic engineering technique that is used to splice together 2 or more DNA fragments that contain complementary sequences. It is used to join DNA pieces containing genes, regulatory sequences, or mutations. The technique enables creation of specific and long DNA constructs. It can also introduce deletions, insertions or point mutations into a DNA sequence. PNAC uses isothermal conditions for amplification, and may be used in living cells. Quantitative Pcr, used to measure the quantity of a target sequence. It quantitatively measures starting amounts of DNA, cDNA, or RNA. Quantitative Pcr is commonly used to determine whether a DNA sequence is present in a sample and the number of its copies in the sample. Quantitative Pcr has a very high degree of precision. Quantitative Pcr methods use fluorescent dyes, such as SYBR green, EVA green or fluorophore containing DNA probes, such as TACMAN, to measure the amount of amplified product in real time. It is also sometimes abbreviated to RT-PCR, but this abbreviation should be used only for reverse transcription PCR. qPCR is the appropriate contractions for quantitative PCR. Reverse transcription Pcr, for amplifying DNA from RNA. Reverse transcriptase reverse transcribes RNA into cDNA, which is then amplified by Pcr. RT-PCR is widely used in expression profiling, to determine the expression of a gene or to identify the sequence of an RNA transcript, including transcription start and termination sites. If the genomic DNA sequence of a gene is known, RT-PCR can be used to map the location of exons and introns in the gene. The 5' end of a gene is typically identified by race PCR. Solid-phase PCR encompasses multiple meanings, including polyny amplification, bridge PCR, conventional solid-phase PCR and enhanced solid-phase PCR. Suicide PCR, typically used in paleogenetics or other studies where avoiding false positives and ensuring the specificity of the amplified fragment is the highest priority. It was originally described in a study to verify the presence of the microbe Yersinia pestis in dental samples obtained from 14th-century graves of people supposedly killed by plague during the medieval Black Death epidemic. The method prescribes the use of any primer combination only once in a PCR, which should never have been used in any positive control PCR reaction, and the primers should always target a genomic region never amplified before in the lab using this or any other set of primers. This ensures that no contaminating DNA from previous PCR reactions is present in the lab, which could otherwise generate false positives. Thermal asymmetric interlaced PCR, for isolation of an unknown sequence flanking a known sequence. Within the known sequence, tail Pcr uses a nested pair of primers with differing annealing temperatures. A degenerate primer is used to amplify in the other direction from the unknown sequence. Touchdown Pcr, a variant of Pcr that aims to reduce non-specific background by gradually lowering the annealing temperature as Pcr cycling progresses. The annealing temperature at the initial cycles is usually a few degrees above the Tm of the primers used, while at the later cycles, it is a few degrees below the primer Tm. The higher temperatures give greater specificity for primer binding, and the lower temperatures permit more efficient amplification from the specific products formed during the initial cycles. Universal fast walking, for genome walking and genetic fingerprinting using a more specific two-sided Pcr than conventional one-sided approaches by virtue of a mechanism involving lariat structure formation. Streamlined derivatives of UFW are lane range, 5-race lane and 3-race lane. History. A 1971 paper in the Journal of Molecular Biology by Klepp and co-workers first described a method using an enzymatic assay to replicate a short DNA template with primers in vitro. However, this early manifestation of the basic Pcr principle did not receive much attention, and the invention of the polymerase chain reaction in 1983 is generally credited to Kary Mullis. When Mullis developed the Pcr in 1983, he was working in Emeryville, California for Cetlus Corporation, one of the first biotechnology companies. There, he was responsible for synthesizing short chains of DNA. Mullis has written that he conceived of Pcr while cruising along the Pacific Coast Highway one night in his car. He was playing in his mind with a new way of analyzing changes in DNA when he realized that he had instead invented a method of amplifying any DNA region through repeated cycles of duplication driven by DNA polymerase. In Scientific American, Mullis summarized the procedure: beginning with a single molecule of the genetic material DNA, the Pcr can generate 100 billion similar molecules in an afternoon. The reaction is easy to execute. It requires no more than a test tube, a few simple reagents, and a source of heat. He was awarded the Nobel Prize in Chemistry in 1993 for his invention, 7 years after he and his colleagues at Cetlus first put his proposal to practice. However, some controversies have remained about the intellectual and practical contributions of other scientists to Mullis' work, and whether he had been the sole inventor of the Pcr principle. At the core of the Pcr method is the use of a suitable DNA polymerase able to withstand the high temperatures of greater than 90 degrees Celsius required for separation of the two DNA strands in the DNA double helix after each replication cycle. The DNA polymerases initially employed for in vitro experiments presaging Pcr were unable to withstand these high temperatures. So the early procedures for DNA replication were very inefficient and time-consuming, and required large amounts of DNA polymerase and continuous handling throughout the process. The discovery in 1976 of Tack Polymerase, a DNA polymerase purified from the thermophilic bacterium, Thermus aquaticus, which naturally lives in hot environments such as hot springs, paved the way for dramatic improvements of the Pcr method. The DNA polymerase isolated from T. aquaticus is stable at high temperatures, remaining active even after DNA denaturation, thus obviating the need to add new DNA polymerase after each cycle. This allowed an automated thermocycler-based process for DNA amplification. Patent disputes. The Pcr technique was patented by Kary Mullis and assigned to Settlers Corporation, where Mullis worked when he invented the technique in 1983. The Tack polymerase enzyme was also covered by patents. There have been several high-profile lawsuits related to the technique, including an unsuccessful lawsuit brought by DuPont. The pharmaceutical company Hoffmann La Roche purchased the rights to the patents in 1992 and currently holds those that are still protected. A related patent battle over the Tack polymerase enzyme is still ongoing in several jurisdictions around the world between Roche and Promger. The legal arguments have extended beyond the lives of the original Pcr and Tack polymerase patents, which expired on March 28, 2005. References. External links: A Guide to Pcr Technology, Select Science, Open Pcr, Open Source Pcr Thermal Cycler Project, U.S. Patent for Pcr, Step through Animation of Pcr, Cold Spring Harbor Laboratory, Open Wetware, What is Pcr Plateau Effect? YouTube tutorial video, History of the Polymerase Chain Reaction from the Smithsonian Institution Archives, 3D models of Pcr equipment for 3D printing on Thingiverse.com, computer exercise. Design of Pcr and Pcr RFLP experiments.